For nearly 20 years, Karen Snover-Clift has been the director of the Plant Disease Diagnostic Clinic of the section of the Plant Pathology and Plant-Microbe Biology at Cornell University. And since 2002, she served as the associate director of the Northeast Plant Diagnostics Network. As director of the clinic, she is responsible for diagnosing plant disease on a wide range of ornamental, food and fiber crops. As associate director of the Northeast Plant Diagnostic Network, she supports the overall mission of providing detection of a possible bioterrorist attack of the nation's natural and agricultural systems, and leads the network's efforts in implementing a laboratory accreditation program known as STAR-D, the System for Timely, Accurate, and Reliable Diagnostics. This morning, Karen will present the pathogen's biology and procedures for diagnosis of the disease. So let's give a warm welcome to Karen, please.
This is a microphone. Let's get that all set up there. Can everyone hear me in the back all right? Great. Sounds good. So thank you all for coming today. Wilt is kind of a newer topic for us here in New York State, and what I'd like to do today is just introduce a little bit of background of how we came to where we are today and some of the work that we do in the diagnostic lab. Some of you may have seen out on the table, we have a couple of items, and you may have seen these in other places as well, but we have this really nice Bolo postcard. Please pick up as many of those as you like and share the information. There's a lot of contact information on the back for people. You might need your reading glasses. It's a little small, but there's a lot of information on these cards. And also the Testo guest brochure is specifically about the clinic and how to submit samples to us, and also some of the other laboratories at Cornell, and helps walk you through that process. We also have a very specific sample submission form for the Wilt samples, and we have lots of copies of those with us today too. So if you're interested, these are all on our website, or you could see myself or Marianne Karp, who's walking around with the camera right now. We can get you some of these. So I would like to introduce Marianne. Marianne is a newer employee in the diagnostic clinic. She has been working with us for about a year, and she has done some really incredible things for us for our molecular capacity in the clinic. So Marianne's title is the molecular diagnostician for the clinic, and she has done all the work on Wilt for us. So thank you, Marianne. So let's get started. So Wilt is caused by a fungus, Ceratocystis fagacearum. Here's a map that shows basically where it was prior to our discovery, and it depends on what literature you look at, but around 1942 or 1944, we believe it was first described in the country, and it was really located in those central states. Over time, in the 1990s, it was discovered in Texas as well, and that's a very different system there because they have different trees, and the live oak is their main concern down there. Symptom looks very different on the live oak, but we did spend some time down there not too long ago to learn a little bit about what they are doing with Wilt, and that's been very interesting for us. So we found it for the first time in 2008. In a small area in Schenectady County, approximately 180 miles from any other known infection. So that basically tells us that it wasn't introduced naturally. Probably a man-made problem brought it into that area. It was found for a second time in the same neighborhood in 2013, so 5 years later. So since that time, around the 2013 timeframe, we only had 2 cases, and they were in the same neighborhood. So it was a very localized infection. So just to give you a little bit of background, it is more typically found on the red oak species, so things like red oak, black oak, and pin oak, much more commonly than the white oak species. And we often tell people how they can tell the difference there, that the red oak group has the pointed leaves and the white oak group has the rounded leaves. A lot of people have come to me and said that we understand it doesn't occur on white oak, and that's not actually true. It can occur on white oak, it's just not as severe, and often it will take many years to kill a tree if it does end up killing the tree. So the red oak is what we're really concerned with, because that infection is very quick. So just a little bit about northern red oak. I know I'm preaching to the choir here about how important it is, but we use red oak for a lot of things, right? So lumber, furniture, cabinets, paneling, flooring, caskets, unfortunately. And it's got a very high value for excellent firewood, and as well as for the environment, right? So the acorns are used, the trees are used for shelter for many animals, birds. And I love this little fact that a single turkey can consume 221 acorns at a single feeding. So how does the tree actually become damaged by this organism? Well, the pathogen gets into the vascular system of the tree, and it basically plugs up that system. So if you know things like Dutch elm disease and chestnut blight, it's a very similar pathogen and a very similar mode of action for damaging the tree. And the tree basically can't feed itself any longer because it cannot move the nutrients it needs back and forth. Here's a nice picture of some of the leaves that we found not that long ago. I think it was 2016 for those pictures. Typically we see that marginal leaf scorch, but a lot of other things can cause that marginal leaf scorch. There's environmental factors, there's other diseases. So just because you're seeing that doesn't mean that oak wilt is there. So we look for that marginal scorch, we look for banding of maybe yellow and brown discoloration. The leaves can appear water-soaked or wilted. The leaf color may turn like a dull green, and one of the big things is that the leaves can fall to the ground quickly, all colors of leaves. So if you do approach a tree and you look down and you see green, yellow, and brown leaves all on the ground, that is an indication that oak wilt may be there. It would be definitely a good candidate to sample. So when those vessels get plugged, it does cause a discoloration in that tissue, and we've often referred to that as a streaking, and we've learned a lot recently, but with Jenny Jeswick, who has taken us out looking at some of these trees and giving us guidance on exactly what to look for in that streaking. So we've learned quite a bit, and we know that it's not always this typical streaking that we would see, like say with Dutch elm disease, where you have that real brown discoloration. It can be more of a grayish, bluish smear that we're actually looking for. Another symptom that we see is these fungal mats. These are very diagnostic. If you've ever smelled one, you'll remember it, and it really helps you when you're looking at other trees. It helps us in the laboratory when we're culturing, because when you open that plate, the smell is very, very characteristic of oak wilt. Some people describe it as yeast, like a bread yeast smell. I think of stale beer when I smell it, so I guess it just depends on our background. Too much beer in my life. But these fungal pads can grow underneath the bark and cause the bark to split open, and that's where you don't want to actually look for these. You could cut away at where that one, let me see if I got a pointer here, where you see this crack here, and underneath you may see these fungal pads. So here you can see that where they pulled that back, but it really does have a very sweet odor, very characteristic for this disease. So what about timing? So we think the infections take place from April to June. Wilt may occur as early as June or July, and some trees can wilt completely in as little as 3 weeks. That's very quick for a pathogen, so that does concern us. But when we do hear people say, well, I've had a tree in my backyard that's been declining for years, that's probably not a good candidate for us to sample. It's much more likely we want that faster decline for good candidates. We see flagging branches, again, how we see things in Dutch elm disease. Often flagging branches are associated with this, and the red oak definitely decline more quickly than the white oak, which is a much slower decline. So we receive samples pretty much throughout the growing season, but we feel like we really have been getting a lot in the August through September timeframe when people are really seeing symptoms more strongly. So even though this infection can take place early in the year, so far we've been getting a lot of samples about August, September timeframe. But again, just you have to remember, this is a very new thing for us, and it's hard to talk about patterns when we have a very small sample set. So we're learning every year, every month as we're doing this. It is spread primarily two ways. One way is by beetle transmission. Those beetles are very attracted to those fungal pads. So they'll go in, they'll feed on the fungal pads, they'll get the fungus all over their body, and then they'll go feed on another tree, a healthy tree, and they introduce the fungus that way. So that's one mode of transmission, and we kind of feel that's, excuse me, that's the primary mode that we're seeing right now, is the beetle transmission. They also spread through root grafts. So these root grafts naturally form between same species and sometimes with many species of trees, and it's a great way for trees to help each other out in times of stress, but also these pathogens can transmit that way as well. So that's a very common way of transmission in established areas. So how to minimize that spread is breaking those root grafts up, and what will often occur is they'll use these vibratory plows or trenching tools to break those root grafts. So if there's an infection site, those root grafts are broken and it will not go outside of that area. And this is a really common thing that's done in a lot of the Midwest states to protect areas. So I love the billboard that is from the pictures of the Minnesota Department of Agriculture. Don't prune in May or June. Prune in May or June. So we feel that that's a really high risk period. That's when the beetles are going to be flying around, they're going to be feeding on those fungal mats, and they will transmit to other trees. And if they're wounded trees, they're more likely to get that infection in place. So that's a really important factor here. We don't want to be pruning during that time unless it's really critical. If it's a natural break from weather, wind, whatever, we do recommend covering that area with either paint or wound dressing. So if you're pruning, or even the accidental wounds. A lower risk period is from July to October, and we suggest that covering those wounds at that time is kind of optional for you. If it's a place where we know oak wilt has occurred, I would strongly recommend it. And then the so-called safe period is November through March. And that's really the preferred time to do pruning. We started giving a lot of these talks last year and this year, and I've actually had people that attended presentations call me and say, I've got a team lined up to prune next week, and we're in April now, what do I do? And you know, I can't make that call for them, but that is a real high risk period, so I know a number of people have actually changed the timing and put it off until late fall or into the winter. And in infected areas, of course, firewood is an issue. So movement of firewood is not a good thing. If you've picked up the DEC bag outside, you know that says that right on the side of the bag. And we should not be moving firewood. So how do we minimize this spread? Here's just some pictures from where they're protecting trees in infection sites. So debarking or wrapping in plastic or burning and burying prior to the April 1st time frame is really a good idea, so that you dry out those mycelial mats so they become exposed, and hopefully they become inactive. Stumps even should be taken, the bark should be taken off all the way to the ground on stumps as well. And then a lot of places will bring in these portable sawmills, so you're not moving the wood long distances, they actually bring it to the site. And even some of the debris they will cover with black plastic to protect it. So what happened here in New York State? So in 2008, we found it for the first time. I had been receiving samples for oak wilt since the day I walked in the door at the diagnostic clinic. people would send samples and say, this really looks characteristic. And we never had positives. And I didn't really understand why, because they have it in Minnesota and Wisconsin and in Ohio, those central states that have very similar weather conditions and environmental conditions as we do, very similar tree species. So why weren't we getting it here in New York? It was a question, but it seemed to be a barrier that it was not coming into our state. It was in Pennsylvania, but we didn't have any cases very early on. So in 2008, we received another sample, and I thought, well, yeah, we'll look, but it's probably going to be another negative. It's not there. And we were quite surprised when we did get the pathogen in culture for that 2008 sample from Glenville, New York. So we isolated the organism and matched the morphological characteristics to Ceratocystis fagacearum, and then we sent an isolate to a researcher at Iowa State University for confirmation, and he performed the molecular testing, PCR, and sequencing to ensure it was the pathogen, and it was a match. So this is just some pictures from the DEC folks of how they mapped out that neighborhood, and I'm just showing you this as kind of an introduction of what we did, but they'll, I'm sure, talk about this a little bit more later. But helicopters were used to survey the area, and they established some protective zones, some quarantine areas, and a number of trees were removed. They had a cutting plan for what they wanted to do. They actually removed the trees here, and you can see the piles of mulch, the chips. It's kind of a sad situation to see. They did also go in and do quite a bit of trenching to break up those root grafts. So here's a before and after picture. So it is quite devastating when you have to remove these mature trees, even in just one backyard. So 5 years later, we found it again in the same backyard, and this was an interesting situation as well. During this time, all of the foresters, DEC personnel, ag and markets personnel were keeping their eyes open, trying to see if it was in other places and be observant to see if it was there. But we didn't receive any samples that were positive during that 5-year time frame. But we did receive a sample in 2013 that was sent to us for bacterial leaf scorch testing specifically. And that's a very different test, and it uses different plant parts. So we definitely use leaves in that situation, and the petioles of leaves, where in oak wilt we want larger twigs, well, you know, like an inch in diameter size twig. So when the person sent it in and we just received leaves, we tested it. Sandra Jensen in our laboratory tested it, and she got a mild positive for the bacterial leaf scorch pathogen. But just based on what the the person was saying, she didn't really think it matched. So she's very persistent, and that's why we love having Sandra in our laboratory. And she asked the person some more questions, and then she asked for some more plant material because she suspected that it might be oak wilt, especially when the person revealed they were from the neighborhood in Glenville. So they did share more plant material with us, and then we did the same. We were able to isolate it from the tissue that was sent to us, and we sent it off to Iowa State for confirmation molecularly, PCR and sequencing. So this is the neighborhood here. You can see again the before and after is pretty sad, kind of a gloom and doom story, but the reason we're having these presentations is we want to catch it early, right? Catching it really early in the process is key to management and hopefully successful eradication. So because we had this twice now, I pursued a specialty crop block grant so that we could do more work on it. One of the things, because we had it twice, was that I wanted us to implement PCR testing, molecular testing in our laboratory. So we pursued a grant, and the objectives of the grant were to identify favorable sites, and that was DEC. They were a huge partner on this grant. All the funds came to the clinic to do the work, but they did a lot of the groundwork. They identified the sites, they did a lot of the sample collection, so they made a huge contribution to this as well. We did a lot of collecting of samples to determine if the pathogen was anywhere else in the state, and we implemented the PCR in our laboratory. So we wanted to see if we could also get the pathogen directly from tissue. So it was my understanding at the time that a lot of laboratories would try to grow it out in culture, and then from the culture they would do the PCR testing. And we wanted to know if we could do it directly from the twigs to take out that whole step, which is a very long process. So our grant was put in place. We put together some sample submission forms and instructions for how to collect samples. We put together a poster and those BOLO postcards, and we have a new poster. So if you're interested, we brought a few with us, and we'd like to have these hanging up in reception areas and places like that where you think people might read about it and become aware of oak wilt. So if you're interested, see myself or Mary Ann for those. So the test methods that we used, we did isolation attempts. We looked for that tissue that was streaking. We plated it out on acidified potato dextrose agar, and also, which is a very common agar, and also in some cases the Barnett's medium. So that was one that was developed specifically for oak wilt. The incubation can be a pretty long process. We look at them quite regularly, but it can take 30 days before we see anything that is similar to the characteristics we're looking for, and it can even take longer than that sometimes. So we also did DNA extraction on the tissue, as I was just describing. We did it 2 times directly from that tissue, and then we also did it from cultures if we were able to get a characteristic organism growing out on the plates. And then we did molecular analysis. It's a double-nested PCR procedure, which just means it's basically doing a PCR twice. So it's a lot more work for Mary Ann. And then the PCR searches for a section of DNA that says that this is oak wilt, and it replicates that over and over again till it gets to a level that we can recognize it. And then we also sent off material to the Cornell Laboratory for sequencing if we did get a positive with our double-nested PCR. So the sequence would come in, we would get the sequence from the laboratory, and then there's a database of sequences out there, and we blast it against that database. And the database for oak wilt isn't really large, so we would like to contribute to that and add our sequences as well. So that is something that we'll do with a project this year. So we received 22 samples that first year. The number 22 of our 22 was submitted very late in the season from an arborist on Long Island, working with Marjorie Daughtry. And they had tried to determine what was wrong with these. They're fairly young oak trees, which isn't characteristic for oak wilt. But he had kind of ruled out everything else and couldn't figure out what was going on. So he saw some of our materials and talked with Marjorie and suggested, hey, let's send it up to the clinic, see if it's oak wilt. So it came to us, and we had a PCR positive. The molecular test was positive for the trees. They were pretty far gone by that point. The material was very dead. Dead is dead, but very dead. And we were not able to get it in culture from that sample. So the following year, we received 55 samples, and we went back to that site where we had that suspect a number of times, in January, in February, and in April. And as we're learning about this process, we know those aren't really the ideal months to do this type of sampling. So we did not get anything from those samples during those months until April. And then April, we found a small colony growing on a plate with a lot of other stuff growing around it. We were able to retrieve that out of the plate, run PCR, and we received a positive there with PCR and sequencing. But unfortunately, we received a lot of other samples from Long Island that ended up coming up positive. We had in July one from West Islip, which was very close to the original Central Islip site, Little Hogneck area, Riverhead, and eventually Brooklyn as well. And then in September had a couple others from the Central Islip site. So here's the DEC map just showing that across the state, and there shows the 4 locations out on Long Island here, and then the one in Brooklyn. And just a picture that Jerry Carlson shared with me of overview. I just wanted to show this to you because of it, there's a lot of forest in this picture, right? There's a lot of trees around that site. So that's a little scary. And just some pictures of the symptoms that they were seeing there. So a lot of marginal scorch. This is the Brooklyn site, and that's a drone taking a picture, which is just so cool. And that's the tree that they had removed at the Brooklyn site. And then we had a little bit of surprise. So we're working on all these Long Island samples, and Sandra knocks on my door and shuts the door, which is never a good thing when Sandra comes into my office and shuts the door. And she said, well, we got... So this sample was from Canandaigua, and it was a very alert homeowner who had a horticulture background, had seen some of the materials that we were sharing, and she was concerned that maybe her tree was infected with oak wilt. So they submitted a sample actually through the extension process. They took it to their county office and then brought it to our clinic. We tested it, it came up positive, and then the DEC folks went out and collected an official sample and did the whole process over again and got everything a second time. So I'll just show you some pictures here of that site. But the take-home message here is now we've got a third area that, again, is very far away from those other areas. So probably 150, 180 miles from all of these areas, making a triangle. So this is the homeowner's site, again, surrounded by forest. This is the tree. So it had been declining for some time that year. And this is her home. And if you look closely, this is the tree we were just looking at and that tested positive. And on the ground here is a stump of a tree that she had removed the year before. So possibly that tree was infected as well when she had that removed. We're not sure, but it was right next door. And just to show some of the leaves that were down on the ground in this area, these are all Mary Ann's pictures. She went up with the team to do the official sample. And now our new map showing Glenville with the original find, and then the finds on Long Island, and now this one in Canandaigua as well. So what we learned from this project was that getting the organism in pure culture can be an issue, and we're having issues getting these samples from Long Island in pure culture specifically.
Don't know what's happening. It's cutting in and out. I don't know where that.
How's that?
Nope.
Nothing?
Let me go.
Testing. I didn't have it in my pocket that time.
No, no. I have to call somebody else through the door.
Okay.
See, I'll just stand up here.
That's it. And I'm not going to move much. I'm just going to stand here like this as well. So, yeah, I feel very stiff, like I have a neck brace on. So, we did find that it was difficult for us to get it in culture from some of the locations that we were getting samples where we were having the PCR positive. So to us, it was a really good tool that we were including the molecular testing now. Because if we hadn't included the molecular testing, we would may have missed those samples. I know it's not going to be on the tube, but should we just keep going?
Yeah, just keep going.
Okay. It's very weird. So what else we learned was that isolating from November to April can be very difficult, and some of the colleagues that we've talked to have confirmed that they have a hard time doing that as well, and some that we talked to don't even bother trying during those winter months. And that the PCR was confirming this much more often than we were getting it in culture. So again, incorporating those other tools was very valuable for us. And because we were having the difficulty with the cultures and isolating some of these organisms, We also were thinking it would be a really good idea to have a second molecular test. We are doing sequencing, so that's different, and that's helpful. But we really like to have another test that, in the cases when we're not getting the isolates, I really feel so much better when I have multiple tests telling me the same thing, because when I do convey this information to DEC and ag markets, they make some pretty significant decisions based on our testing, and we want to make sure we get it right. So, we put in for another specialty crop block grant, and luckily that was successful, and that's going to begin December 1st, and that's going to be Mary Ann's life this winter. So what she's going to do is see if we can tweak the auger recipes a little bit. See if there's any way that we can minimize the secondary organism growth on some of these plates and really optimize the growth of the pathogen. So we're going to try that out. We're also going to validate another PCR method, a real-time PCR, which hopefully we'll get faster answers because that's what real-time is all about. So we hope to get the faster answers with that, and we have, we have a 96 well system, and our current system is individual tubes. It uses a Cepheid smart cycler as the platform, and those are kind of phasing out now. So we've got a new real-time PCR machine, an ABI Plot Studio 3, sounds good. And Mary Ann's going to validate the protocols on that machine for us. And if there's time, and Mary Ann's so efficient, I'm sure there will be, we hope to try to do a multiplex PCR. So what that is a PCR where you're testing for more than one organism in one tube. So we'd like to test for bacterial leaf scorch at the same time that we're looking for oak wilt, because they do kind of mimic each other at times. So we think that would be a really valuable test as well. And then on top of all that, we want to contribute our sequences to the GenBank database. So new for 2017, we reprinted a bunch of the BOLO cards. So again, please help yourself to those BOLO cards and spread them around. And we put together a new poster. If you're interested and have a place to hang these up where people will read them and be able to learn a little bit about oak wilt, please do that. We tried to make it a type of poster that maybe somebody at a reception desk can look over and just pick up a little tidbit. Say, oh, I didn't realize it was in 3 areas of New York State, or I didn't know that red oaks were more susceptible, and didn't know firewood was an issue. So I'm sure all of you know that's a message that's hard to get across to people. Movement of firewood, they do figure, hey, I've got wood in my backyard, I'll just take that with me. Here's our new sample submission form, and all of this stuff is available on our website as well. And here we are working with a lot of experts from other areas. We've worked with experts from Texas, Minnesota, West Virginia, and we've really learned a lot from all of these colleagues. They are so good about sharing their information. But one thing that we learned from one of our colleagues is he said, don't believe everything you read about oak wilt, because some of the literature isn't always accurate. So really, take it with a grain of salt, think about it as you're reading it, and just know that there is a lot of variation from location to location. So what's going on in Texas might not occur here in New York State. One of the other things that we want to incorporate, and we worked on this a lot with Jenny Jeswick when she came on her first trip to New York. We're lucky to have her a couple times here. Was incorporating trunk and root flare samples. So we hope our protocol for the coming years will be, if a tree is declining this year, we will ask for the twigs, the branch segments, and if the tree has been dead for some time, we'll ask that they also include a root flare or some trunk wood that has the smearing or the streaking. and that streaking issue is there. We want to make sure people understand that it really doesn't look like Dutch elm disease streaking, and that's what we all have in our minds as plant pathologists learning about streaking. I think I'll speak for myself. You think of this kind of brown discoloration, and I think this is a little bit different. So, key conclusions for what we learned. The PCR for us was even more important than we originally thought, because it is showing us some areas that we need to focus on and make sure that those areas aren't infection centers that got missed. The root transmission of the disease might be different for how we see a fetal transmission. So we know a lot of the people we're working with have worked in this field with the root transmission, those root grafts, and just wondering if the streaking, the flagging of the branches might differ quite a bit with just a fetal infection transmission for them. And that our primary focus really needs to be on outreach and education, because this is one that you have to get the sample right. It's not something that someone can just walk out their back door and see some leaves on the ground and send them into the diagnostic clinic for diagnosis. You really have to know what you're looking for. You really have to go to the appropriate areas to collect it. So that's why we've worked so closely with DEC and Jennifer, making sure that we're doing these collections in the right way. And in the coming months, we want to really get some supporting documentation out there, picture clues, things that we can do to really help people collect the proper sample. And the discoloration of that sapwood issue is a big thing for us, so we got to get that message out to people too. There are a lot of other things that can cause discoloration of that bark, and just using a metal tool on the wood can discolor it in a way. So you really have to know the difference between insect feeding damage. Maybe in some of the cases we had herbicides applied to these stumps. That discolors the wood in a way. If you've worked with oak in the past, you know just taking the bark off, it discolors. So that changes. So it's really important to just keep those things in mind as they're collecting samples and know where the best place to collect is from. So I'm going to stop right there. And do we want to answer any questions right now? Or it looks like, yeah, we got a couple questions, so let's do a couple right now.
I think you've given an excellent presentation. I think you need to call up all the biology teachers in Nassau and Suffolk County and give a discussion with all the high school students and get that out in the field to start looking for this stuff. Your brochures should all go to every high school biology teacher in Nassau and Suffolk County, so you have plenty of boots on the ground to look for this stuff, because we can't, the people in this room can't do it ourselves. And I believe there's infested trees up and down the Wantagh Parkway, and somebody needs to check it out.
Thank you. Yes, I know that Jennifer has a hotline, DEC has a hotline where people call in and can say where trees are damaged to give them a heads up. And there are so many, and this year we're seeing a lot of damage by two-line chestnut borer. So a lot of that is happening, and I know Jenny had said something about seeing this much in her area, that we're just inundated with this damage and seeing the tunneling being so extensive. So there are a lot of other things, and I really appreciate your comments, because it's very interesting to get those eyes and ears out there, and kids do get very interested in this, so it is a great way of introducing plant pathology to them and have them keep their eyes open for us. We definitely need the boots on the ground.
Karen, can you explain what's actually going on in your incubation period in the media? You refer to in your sample as contaminants, which are really other organisms.
Yeah.
What are you likely to find there, and how do you tease that out?
Yeah. That's difficult.
Yeah.
So the auger medium that we use, acidified potato dextrose auger, has a little acid in it, so that kind of keeps some of the contaminants out of there. But the wood itself has a lot of secondary things already growing on it. We're very careful not to let our collections fall on the forest floor when we're taking wood collections, the wood chunks, because we don't want to take more organisms with us. So, and we do surface sterilize. So what we'll do is spray it with some ethanol and flame it off, so we get that initial stuff off there. So the organism is deeper in the wood, so we should get that and not those secondary things that are just on the surface. There's so much stuff in the air, so we just have to be careful that we do everything in a controlled environment, a very clean environment. Marianne is very good at sterile technique, and if anybody can keep the organisms out of there, she can. But they do feed on that plate just like anything else. So if they're on the plate, they're going to grow. And the pathogen, from my understanding, is the pathogen is really, really active in the tree, and that's its ideal environment. It can grow on this artificial medium, but not as well as it does in a tree. So some of those other things do outcompete it for space. And we just have to watch it closely as it's growing out. And if it looks characteristic, grab it, do a subculture before it overgrows with other, yeah.
You ever track beetles with this?
We haven't, but that is something that I know DEC has discussed, because we could just grab a beetle, grind it up, and run the PCR and see if it's on, the fungus is on the beetle itself. So that would be a very interesting project to incorporate.
You mentioned it briefly, but in one of your samples you said that you found bacterial leaf spores, so
Yes.
they can be concurrent on the same tree?
Absolutely, yeah, you can have both.
Can you describe the differences in appearance between root and beetle transmission? The trees affected by either?
Yeah, I'm not sure if we know very distinct differences just yet because we have such a small sample set. But I'm thinking because the beetle infection is typically in one location, you would probably see localized streaking versus streaking throughout the entire system. Where the root infection, I would expect to see more consistent streaking because it's coming in through those root grafts, and the beetle is kind of a localized area.
And for those trees where you had root grafts, you may want basal samples taken rather than from the
Absolutely,
branches.
yeah. In Texas, because it's a totally different thing in Texas, it's live oak. They have very different symptoms on the leaves. They have a banal necrosis, which is not what we have here, and they have a half-leaf symptom that they look for. But they take those root flare samples from every sample.
So,
because they have been very successful in getting it out of the root flare.
Yeah, so where you take the sample might be influenced by how you think the tree might have been infected, either through root or beetle.
I believe so, yeah, I think so.
I'm curious about the incidents of all time.
Yes.
Have you had done any forensic pathology to find out how it got someone dispersed?
Not yet. No, we haven't done that yet. We've been basically trying to identify where it is. We've had a lot of negative samples in between these positives, so it's not like we just did those samples and they were all positive. There are a lot of negatives coming through as well. And it will be interesting to map it out this year because we have a much bigger sample set this year. But we haven't done any kind of forensics looking at if there's any variation in our isolate to West Virginia, Minnesota, Texas. How does that vary? But that would be a very good next step.
In the process of breaking the root grafts, you are in a sense breaking the roots. There's no
Yes.
question there.
Right.
Does breaking the roots weaken the tree to make it more susceptible?
I don't know if I've ever heard of any problems with the root graft. Anytime you have a wound, of course, you can introduce other things, opportunistic organisms, but I haven't heard that people have stopped that because of other pathogens. I think it's still a very viable resource in areas where oak wilt is prominent. Oh, what did you say first?
How reliable, Karen, is the PCR if both bacterial leaf spores and oak wilt are indicated in the results to be isolated as oak wilt?
The PCR itself?
Yeah, if that comes back and then both results
Right.
suggest
Yeah.
bacterial leaf scorch and oak wilt.
Yeah. So actually, we're not doing PCR for the bacterial leaf scorch. We do an ELISA test.
Right.
It's a serological test. So they're actually right now 2 separate tests. We hope to put them together in a multiplex. So we do the ELISA separately. That's a commercially available kit that has validation done at the company, so we feel very good about that. And the Oak-Wilt procedure, we're following the procedures that a number of other researchers have already used and have validated. So we're following their procedures. We've done it in our laboratory using positive controls to make sure that we're doing it correctly. And then doing the sequence on top of that helps us validate that as well.
But
because then we're getting the organism based on the sequence that we're getting back.
For management practices, if a PCR comes back positive and it's a group of red oaks, but it's not been in culture yet, are you confident enough to go on the PCR to say it's good enough to go on that?
That's not our call, but DEC says yes.
Okay.
They're willing to accept the molecular testing as confirmation.
Okay. Thank you.
Yeah. So of course we want to do as many tests as possible, and we really try to get as much in culture as possible so that we have all those answers. You know, we don't want to get this wrong. We want to make sure that we're confident in our answers. So we work really hard to make sure that those tests are accurate and run the test multiple times at times to make sure. It is difficult because a sample, when you have a sample, even when you break it in half and say, we do a test and Marjorie does the test, we can get different answers. It's not the same sample. Even though it's the same chunk of wood cutting it in half, it's not the same sample. So you can have variation there, and that's hard, I think, for some people to understand. And so far, we have such a small sample set here in New York, it's hard to make generalizations on what we're actually seeing. And I'm confused why we're not getting cultures on Long Island. And so is Marjorie, I believe. And we're doing our best to try to figure that out. That sums it up, kind of.
Number one, when you sever the roots, it has to be root-to-root transmission, so there's no soil-borne pathogen that can infect the roots after severance.
There could be. Yeah, we don't know if there's any there, but
as far as sampling,
right.
As an arborist, I know that this year was a very wet year. We had a lot of things going on.
Yes.
But there's been a lot of discussion as to whether or not we're seeing Oak-Wilt. And I can tell you, on my part, we're kind of holding off to see what the samples are, whether we have bacterial scorch. It was a bad year for fungal and bacterial things across the board this year.
Yeah.
But maybe we'll just gather a lot more samples for you. The discussion's out here as to what we're looking at. Are you seeing it? We think we see it along parkways, but we're not sure.
Right.
So part of that might be if you want us to send more samples, we can do that.
Right.
Part of it is we just don't want to overwhelm you.
Right.
And we're still discussing as to what we're seeing.
We want to really focus in on those samples that are the best characteristics so that we can concentrate on that. Calling the hotline and alerting DEC to those sites is great because they're gathering that information. And a lot of the stuff that Jennifer and I have seen when we're driving around, and I believe Marjorie and a couple of our experts too, is that two-line chestnut borer is extensive this year. So if you can cut into some wood and look for that, that could make you feel a little bit better about, I'm really leaning towards a two-line chestnut borer here, because it has been a very bad year for that.
But it has been a discussion this year. Every arborist I talked to is seeing what we think is symptoms.
Yeah.
We're not sure. It was a fungus year.
Right.
So there's kind of a holding pattern for us. We don't know what we're looking at.
Exactly, and we're trying our best to get you materials, right? So we've collected a lot of pictures. Mary Ann takes pictures of every sample that comes in, and we have also tested a lot for bacterial leaf scorch. Rob has given us the okay to do that. So that's a learning experience for us too, because we want to know, okay, we saw this marginal scorch. It didn't look like Oak-Wilt to me, but was it bacterial leaf scorch? It'd be nice to know that. And we have gotten quite a few positives back on some of the samples that we collected. So I wish we could get you information faster, but I think it's a process, right? So we're learning a lot throughout this process.
Nice job, Aaron.
Thank you.
I think one thing we need to think about is red oak is extremely susceptible. But there's lots of different kinds of red oak. And
Yeah,
simple
right.
development in different red oaks can be different.
Right.
So,
Absolutely. So the majority of our positives have been true red oak, northern red oak. But we did have, I believe, a black oak and one white oak. So it's interesting.
And the first was Kim.
And the first was pin, right? That's right. Yeah.
Two quick questions. Did I see on your map you had located a population of this in Southwold?
Yes. Yeah. Is it, what town is that? It's
I'm sorry,
Southwold.
what's that?
The Southold positive, is it the town of Southold?
It's in the town of
It is
Southold.
town of
Yeah.
Southold.
And also, has it been located in Scarlet Oak yet?
I don't think so.
I don't think so.
I don't remember off the top of my head.
No, I don't think so either. I think it's just the black pin and white.
Did we just have one last question? Cool.
I just have a question about
One
graft.
last.
When you develop the trench line to break up the grafted roots, do you have, you have to stay out of the drip line, or how do you determine, the lines between the trees, what's a safe place to kind of do that?
Yeah, I think that's a call on the arborist part, and sometimes property line as well. They have to talk to neighbors, and as you can imagine, trenching doesn't work everywhere, as Joe will comment on. The cemetery, they can't do trenching. They can't dig down at the cemetery, so it's not a good thing, right? So that doesn't always work, but I think a lot of it is really property lines, homeowners, but they try to find a good site far enough away from the tree that they're not right underneath it, but that it's a protective area between it and the next property or the next natural area.